I feel satisfied, of course, for the patients. And I think this is also a remarkable example of the synergy that is possible between clinicians and basic researchers. The thing with this story about this form of leukemia is quite atypical in that the treatment preceded the discovery. So it's not that the discovery led to the development of a treatment, but the discovery led to the understanding of the disease and how the treatment works. So things happened in a totally unexpected manner while I was studying the role of the hepatitis B virus in liver cancer. And that's the way I discovered a new gene, which was mutated, altered in this liver cancer. And we then identified this gene as the gene for the receptor for retinoic acid, which is the active derivative of vitamin A. So we then moved to this form of leukemia precisely because retinoic acid can treat 75% of the patients suffering from this leukemia. So we thought that there might be a link between the leukemia, the treatment, and retinoic acid. So what we discovered is that in these patients, in the leukemic cells of these patients, 2 chromosomes had broken and then fused abnormally to each other to give rise to a chimeric protein, a hybrid protein with half of a protein, which is called PML, and the other half from another protein, which is the receptor for retinoic acid. And this chimeric protein was the faulty protein responsible for the leukemia. And through the identification of this faulty protein, at the same time we understood why the treatment is so efficient. It's because it forces the faulty protein to behave normally, to behave like a normal receptor for retinoic acid. And it forces the leukemic cell also to behave normally, to finish its maturation and just do its job normally. And then there was a next chapter. We left the molecule for the cell, and we now discovered a cellular defect associated with this type of leukemia. We discovered very small structures in the nucleus of the cell, and when we looked at the leukemic cells, we found that these structures were totally exploded, disrupted. So it was a defect. And strikingly, retinoic acid treatment was able to restore the formation of these structures, to correct the cellular defect. So retinoic acid was not only able to correct the molecular defect, but also the cellular defect. And then there was a last chapter, which started when Chen Zhu discovered that arsenic, like retinoic acid, was also very efficient to cure this form of leukemia. So we wanted to understand how it works, arsenic. Why does it cure these patients? And we discovered that arsenic, in fact, leads to the attachment of a very small protein onto the back of the faulty protein. And this tagging, this labeling, leads to the degradation of the faulty protein. So just to summarize, on one hand, retinoic acid targets the faulty protein by forcing this protein to behave normally, and on the other hand, arsenic targets the same faulty protein to degrade this protein. So the two compounds function as two different weapons to specifically target the molecular defect, and thus represent a prime example of an oncogene targeted therapy with such a high level of cure. I think basic research is just essential, since it leads to the understanding of the normal cell, and doing so it leads to the understanding of the disease cells. And we absolutely need this type of research driven by the soul, curiosity. And I like to cite Éric Orsena, who is a French writer and a member of the French Academy. He says, do you know that the word curiosity comes from Latin, cura, which means care. So be proud of this fault. To be curious means to take care. And I think this summarizes pretty well the necessity of basic research. That is freedom, pure curiosity, and this is the way we will find clinical application. If you dictate to a scientist to find a treatment, it will not work, or very few chance it will work.
Looking at your research today, where's your focus?
Now we are working on an intriguing type of modification of the proteins by a very, very small protein, which is paradoxically called SUMO. In fact, and this tagging, this labeling by this very little protein will drive the proteins towards different destinies. Because to increase the diversity of proteins repertoire, you have the genes. The genes make proteins, but the proteins can then be modified by little chemical group or by little or small protein. And this tagging gives different function to the protein. So we are interested in this modification by this small protein called SUMO, and particularly its role in cancer, because we and other labs found that this modification is hyperactivated in the vast majority of human cancers. And it is hyperactivated because the cancer cell is stressed. And this modification becomes really active to defend the stressed cell. So it's a kind of defense of the cancer cell. So what we and many others and pharma company wants to do is to decrease the level of this modification so that the cancer cell will not be able to survive. And we came on that through the leukemia, because we found this modification affects the faulty protein of the leukemia and is responsible for the targeting to degradation. So it's a long story for the last 30 years with different chapters, but was a fascinating story.